So how's everybody doing this afternoon?
Really great.
Doing all right? Well, it's my pleasure to introduce Jut today. I'm the co-chair of Jut's doctoral committee, along with Dr. Brett Dixon, who, and I want to make sure I get the name of this right, is the director of the Lab of Landscape Ecology and Conservation Biology here at NAU. I'm the director of the Colorado Plateau Biodiversity Center, and I've been really happy to work with Jut for the last, how many years, I don't even know.
Been quite a while.
On his excellent work in cave biology. Also here are his other distinguished committee members, starting with Dr. Frank Howarth, who spent most of his career being a research entomologist at the Bishop Museum in Hawaii, and who is also a very major figure in cave biology, and particularly cave arthropods in the Polynesian part of the world. And so he's really lent some very essential expertise to Jut's work. We also have Dr. Tom Sisk, who is a professor of ecology here and also the Olahos Goslau Chair of Environmental Science and Policy here at NAU. And Jeff Foster, who is a pathoecologist in the Paul Keim Lab. And you might ask, well, why a pathoecologist? Well, Jeff, in addition to helping Jut plan out future genetics research related to what he's going to be telling you about today, has also worked in the Hawaiian Islands on birds and their parasites, among many other things. So I would just like to take a few moments. I don't want to take much time away because Jut has a lot to tell you about today. But I'm proud to say that Jut Wynne is a true bugger. He has dedicated his life to understanding cave arthropods. And I use that term bugger in the purely arthropodological sense. And there's a lot of fun exploring caves, finding new organisms, and doing that sort of work, but it also requires a lot of time in this type of position, looking through scopes at minute morphological details of insects and recording lots of data, and then, as we've been prodding him to do, analyzing all of that data. Jut has always been kind of at the social forefront as well as at the science forefront of thinking. This is a Jut in, which grade was this? In sixth grade. And he's got a very social and enthusiastic personality that has really helped him in his cave work, because it takes teams of folks to go out, carry all the equipment, explore caves, and do that work successfully. And so his enthusiasm has really carried broadly, and that has helped him a lot in doing this work. His social enthusiasm has also carried over to the bugs that he's discovered himself. So this is him giving a high five to a new genus of cricket that he discovered in Almalpais, I believe. Jut has been actively involved with a number of different cave associations. He's been a fellow with the Explorers Club and a fellow with the World Explorers Bureau. And so he has done a lot of work not only to do this work, but to share it with the broader audience. And if you look at his website, you can see a lot of media in which he is doing that. But also he's been an invited speaker in many conference and university settings to tell people about this exciting work. And it doesn't end there. Jut has 47 papers and technical reports in publication. 15 of those are in peer-reviewed journals, and 7 additional manuscripts are in prep, 2 of which have recently been accepted, including one in bioscience. So and that's the story that he's going to end with today about the insects in caves on Rapa Nui. So he's done a good job both publicly and within the scientific community, of telling the story of his research. Jut came to NAU after receiving a Bachelor of Science from Georgia Southern University in 1993. He also received the Certificate of Echotechnie at the Vrije University of Brussels in 1998. He received his master's degree here at NAU in 2003 and received the award for Most Promising Graduate Student Researcher here at NAU in 2011. So I'm very proud to present to you Jut Wynne and the wonderful research that he's been doing. Yeah, you need that.
All right. Well, thank you so much, everyone, for taking the time out of your busy schedules to be with me today. And thank you very much, Stefan, for that very warm introduction. What I'm going to be providing today is essentially a snapshot of 3 or 4 manuscripts that I've completed related to my dissertation. And I also hope to provide a glimpse into the fascinating world of cave biology. I'm going to be giving this talk in 4 different sections, the first of which I'll be providing an overview of the cave environment, which is going to include the discovery of several new species and genera. And then I'm going to be discussing 3 of 4 dissertation chapters, the first of which is looking at advancing systematic techniques for sampling cave-dwelling arthropods. I'll then be discussing how we elucidated those environmental factors driving biological diversity of cave-dwelling arthropods. And finally, discussing cave fauna as relics of global environmental disturbances. First, this is an overview of the zonal cave environment. It's broken down generally into 4 different zones. The first 2 are photic, the light and twilight. The transition and deep zone are aphotic zones. When photosynthesis does occur in the cave environment, it obviously will occur within this area. Climate is going to be more reflective of surface conditions within the light and twilight zone, becoming more and more stable as we reach the deep zone. And also, as we would expect, we're going to have animals selecting habitat along this zonal gradient. So I'm going to be discussing 4 cave-specific functional groups, the first of which are 2 types of animals that occur within both the deep and transition zones of caves. The first of which we call troglodytes. Now, troglodytes, these are animals that complete their entire life cycle underground and have essentially adapted or evolved to living underground specifically. Characters typical of cave-adapted animals: these animals are depigmented, they have gracilization and elongation of appendages and antennae, their eyes are either lacking or reduced, and they're covered in setae or covered in hair. The second type of cave functional group is called a stagobite, and these animals are simply just aquatic troglodytes. The third functional group are what we call troglazines. Now, these animals require caves during at least a portion of their life cycle. 2 primary examples of these animals are bats and cave crickets. And these type of animals will use caves during the day diurnally and will exit the cave at night to forage. Our final group that we're going to be discussing are called troglophiles. Now, these animals are found throughout the length of the cave. These animals are facultative cave users, meaning that they can complete their life cycle underground, but they can also complete their life cycle in similar surface environments. And this is where it all began for us when we started studying cave arthropods in the American Southwest. This proved to be a new species and new genus. This was from work on North Rim Grand Canyon, and we made this discovery in 2006. And at that point, colleagues and I knew that we were onto something. We thought that because these areas have not been intensively sampled, that we stood a great chance of just making a lot more new species discoveries and potentially contributing significantly to the natural history of these different areas. And our hunch proved correct. Over the past decade, we have discovered nearly 50 new species, 3 new genera of cave-dwelling arthropods from the American Southwest and from Easter Island. And I want to talk about one new genus specifically here. We assigned the name Praetherodesmus to this new genus of Macrosternidae millipede, and this was named after our friend and colleague John Prather, who passed away about 10 years ago. So we did this to honor both him and his family. And as we would expect with the discovery of new species, there's often going to be management implications associated with that. And clearly caves are no exception. And this is because cave-restricted animals in particular are often narrow-range endemics, meaning that they may occur in one cave or one region and nowhere else on the planet. And also, caves have been identified as hotspots of endemism and biological diversity. And because we know that the global footprint of human activities is omnipresent, impacts to caves are no exception. There are several anthropogenic threats to cave biology, the first of which are biological invasions from non-native invasive species. Also, we know that bats deliver nutrients to caves via an allocthenous mechanism. And as a result, the allocthenous delivery of nutrients to caves provide this important nutrient resource for cave-dwelling arthropods. So any disruption to those populations could adversely affect cave populations as well. Deforestation, this is somewhat of a no-brainer. We clear the forest over a cave. It's going to affect sedimentation into the cave and could potentially disrupt the delivery of vegetation into the cave as well, which cave-dwelling arthropods are using as an allocthenous nutrient resource as well. And finally, recreational cave use. The more that people recreationally use a cave, this can lead to further soil compaction, which may render suitable habitat for arthropods unsuitable. So for the first section of my dissertation talk, when we were developing this approach to sampling cave-dwelling arthropods, we wanted to make these across-cave comparisons when we were conducting these different methods, applying these different methods. And the way in which we selected the different techniques that we were using for sampling was by going to the literature, and we identified 7 primary techniques for sampling cave-dwelling arthropods. Now, when we did this, we realized that when we conducted this literature review, that we observed that there was not a systematic approach that was applied over these different studies. So this made inter-cave comparisons difficult. But that's not without good reason. Sampling caves are incredibly challenging. Often they're logistically difficult to access. These can involve a full-blown expedition, which requires portage of sampling gear, climbing gear, camping gear, food and water into the backcountry. Also, caves are both spatially and temporally heterogeneous. And by spatially heterogeneous, I mean we know that, for example, bats aren't going to deposit nutrients uniformly from the entrance all the way to the back of the cave. This is going to be patchily distributed. And by temporal heterogeneity, we also know that bats are going to use caves seasonally, in most cases, or in many cases rather. So there's going to be this pulsed nutrient input into the cave. So as a result, this systematic approach to sampling caves may indeed be quite challenging. But I do think that it's important, as both an ecologist and a conservation biologist, that we strive towards a systematic sampling paradigm. So the objectives of this study were to first determine the most efficient techniques which render the most complete representation of the cave communities sampled. And through this, we were developing and applying a systematic approach for sampling caves, which enabled us to draw these comparisons. And finally, it was our hope that through this work, at bare minimum, we hope to contribute to a fruitful debate regarding how best to move towards a systematic sampling paradigm of terrestrial cave-dwelling arthropods. These are our 3 study areas. Our first study area was in Grand Canyon Parashot National Monument in northwestern Arizona. Caves within this area range from low elevation Mojave scrub to mid-elevation juniper scrublands. In El Malpais National Monument in west central New Mexico, caves were alpine caves occurring at 7,000 plus elevation. And because we wanted to compare these southwest sites to something completely different, we also studied caves in Rapa Nui, which is 6,000 miles to the south of the southwestern U.S. Now, caves within this region occurred at about 1,000 feet elevation and occurred within a largely invasive vegetation community comprised primarily of grasses and guava scrub. And for these study regions combined, we had a total of 26 caves that we sampled. Now, for this, what we did is we did this across method comparison. And we looked at 3 different parameters for this across method comparison. The first of which was observed richness. And this was just simply the total number of observed species per technique per cave. The second parameter that we examined was efficiency. And we simply took the total number of observed species per technique and divided that by the number of samples per technique per cave. And finally was this parameter of completeness. And this is essentially the percentage of the actual or true community occurring within a given cave. And the way that we did this was by taking the total observed number of species per technique, and we divided it by the predicted richness value, which we derived using a program called Estimate S. Now, for sampling our arthropods, as I mentioned earlier, there were 7 primary techniques that were used, that we identified in the literature for sampling cave-dwelling arthropod communities. We selected 6 of those for this work. This is a list of them here. And when we were applying the sampling protocol, one of the questions that struck us from the literature was that we encountered this process of opportunistic sampling quite frequently in the literature. And we also noticed that it wasn't always clearly defined. So a question that we had is this process of this random walk through a cave to collect arthropods, is this comparable or better or worse than a systematic interval sampling approach? And that was one of our questions here. So the interval sampling approach was this. We essentially laid out a grid, and our sampling interval was established by taking 10% of the cave length. So if we had a 1,000 meter cave, our sampling interval was every 100 meters. And then at each one of these sampling intervals, we deployed 3 stations where we conducted timed searches and deployed pitfall traps. And as we deployed pitfall traps and retrieved those traps, we conducted these opportunistic searches where we had 3 observers walking along these transects, as you see here, collecting arthropods as they were encountered. The next approach that we applied was sampling cave deep zones, and we know that these are the areas where cave-specialized animals are most likely to be encountered. The first technique we used is called direct intuitive searching, where we deployed a 1 meter sampling grid, and 1 observer spent 20 minutes time searching within this grid for cave-adapted arthropods. And once that was completed, we deployed a series of baits for several days to then attract arthropods as well, and then we came back at the end of that sampling period to see what we found. Also, as I mentioned earlier, caves are highly heterogeneous. So not all caves will have the same suite of nutrient resource sites. Some will have none. So we also added this additional element of direct intuitive searching to these different nutrient resource sites. And we had 3 different types of nutrient resource sites: fern moss gardens in our Rapa Nui caves, moss gardens in our El Malpais caves, and finally we had these tree root curtains which occurred in El Malpais caves as well. Now, regarding the completeness parameter that I mentioned earlier, I want to spend a little bit more time explaining that. We use this program called Estimate S, and what this provides us with are what we call these asymptotic species richness estimators. And this is an example of a species accumulation curve. On the y-axis, we have the number of species. On the x-axis, we have the number of samples. And when as we increase our sampling effort, we detect those most common species first, detecting those more rare, and then ultimately we reach this asymptote where it doesn't matter how much more time we spend, we're not going to detect any more species because we've detected all the species in the community. Now, this is largely a theoretical construct, but this is how Estimate S operates. It predicts where those asymptotes are going to be. So we selected our best species richness estimator using 2 criteria, the first of which would be this asymptotic-esque behavior of that species richness estimator curve, and then finally a standard deviation of 2 or less. Then for this across cave comparison, we used a non-parametric Wilcoxon signed rank test. And then we wanted to look at how similar or dissimilar these different techniques were at detecting the community that we were studying per cave. And we used a Sorensen similarity coefficient to do that. And finally, we wanted to see how well each individual technique maximized species detection. So we conducted that examination as well. And finally, what we wanted to do was to see overall how well did we sample each study region. So we visually examined species accumulation curves both for individual techniques per study region and the all techniques combined for each study region. Now these are the results of our paired comparisons across methods for the Wilcoxon signed rank tests. We have here in the rows those three parameters that I mentioned earlier: observed richness, efficiency, and completeness. And first I'd like to point out that in most cases for these across method comparisons, when looking at individual methods, they were largely similar. I would like to point out though that question that we had to begin with was, is opportunistic collecting significantly different from this interval sampling approach? And we found that to be the case. In two cases, for observed richness and for completeness, we found those to be significantly different. Turning our attention to the Sorensen similarity indices, for the Rapa Nui study area, we found that there were 54% similarity between searching in those fern moss gardens and that interval sampling approach, and 47% similarity when we were looking at opportunistic collecting to interval sampling. Things were quite a bit more different when we were looking at the southwestern study regions. We had 80% similarity between opportunistic and interval sampling, and about the same, 75% similarity for opportunistic and interval sampling. And now when we're looking at the detection of how these different techniques differentially detect species, I'm using this as an example. This is management concern species, and there were three different types of management concern species that we're examining here. The first of which are simply new species, which, as I mentioned earlier, may have conservation implications associated with them. The second would be newly cataloged invasive species to the island of Rapa Nui. And then for the southwestern cave sites, range expansions, detections that represented range expansions of a given arthropod species. And what we found here was quite fascinating, is that in most cases, individual techniques uniquely identified management concern species. So what this is telling us is had we omitted any of these techniques, we would have sacrificed completeness. For our new species, nearly 60% of those animals, which were either cave adapted or cave restricted, were detected by a single technique. Regarding other management concern species, 50% of those were detected using a single technique. Now these are the results of our species accumulation curves, and what we did is we combined all techniques per study region. This is the very long blue line, and then the shorter lines are the individual techniques per study region. But what's most important to point out here is that in all cases, none of these species accumulation curves neared an asymptote, suggesting that although we applied the systematic framework, we intensively sampled these sites, we still have more work to do to most completely detect all the animals represented within a given study region. So in conclusion, multiple techniques do indeed maximize completeness. We see here that, and the prime example is that those management concern species, most of them were detected by applying a single technique. We also found that deep zones in those nutrient resource sites were critically important to sample because most 11 of 18 cave-adapted or cave-restricted animals were detected within those habitat types. And finally, the application of multiple techniques, and moreover to add this multiple site visit component to visit a cave as many times as possible, will ultimately be required to maximize completeness of sampling. The third part of this talk, we're going to be elucidating. I will be talking about how we elucidated the environmental variables driving biological diversity at two scales. The first scale is the regional scale, and by that I mean the southwestern U.S. study sites, the caves in El Malpais and Parashot National Monument. And then we looked at this from a broader scale analysis by adding the Rapa Nui data set. But first, let's discuss our understanding of what drives diversity in surface ecosystems. Well, we know that it's the abiotic and the biotic factors that drive biodiversity patterns. And these are three of the primary explanations for that, the first of which is the latitudinal gradient hypothesis. And this has been demonstrated in numerous studies that we have the most diversity along the equatorial belt, and the diversity of life decreases as we move north and south towards the poles. The other idea is the biological productivity hypothesis, which states as you increase productivity, photosynthesis-driven productivity, because it's on the surface obviously, that you're going to increase biomass, and by extension you're going to most likely increase the number of species within a given area. And finally is our species-area relationships. Now this relationship has been demonstrated in just about every single study where it's been examined, and that is the larger the area, the more species that are likely to occur. Now caves are similar, but there are some nuances. The first of which, rather than this latitudinal gradient, there's actually a zonal gradient. And we know that richness is greatest near the entrance of the cave, and it decreases as we go towards the back, towards the deep zone of the cave. And regarding productivity, in most cases there's not photosynthesis. And we already mentioned that there are exceptions where there's photosynthesis in the entrances, but in most cases it's decomposition driven. And that is by delivery of these alloctonous resources into the cave, bat guano, flood detritus. And if we have the appropriate temperature and humidity, or moisture rather, the more moisture, the greater the decomposition, the greater the available energy, the cooler, the opposite. Also, the species-area relationship has been demonstrated in at least 2 studies that we're aware of, and this was a positive relationship between cave length and the number of cave-adapted animals. So our objectives of this study were twofold. First, we wanted to identify the most important drivers of biological diversity at the regional level, that southwestern regional level. And then we wanted to add that Rapa Nui data set and see if those drivers were consistent at this broader scale. As for field sampling, we applied the same systematic protocol that I discussed in section 2. And as for analytical techniques, we developed our arthropod environment relationship models using GLMMs, which are general linearized mixed models. And we developed a suite of these models which we used to identify the relationships between those environmental variables and biological diversity. And then we used a model selection process where we used a backward stepwise approach, and then an AIC value of 4 or greater that provides us a level of strength regarding the differences between those models, and then also AIC weights, which is essentially just the percentage of weight or strength of evidence for a given model. For example, 0.1 would be an excellent model, 0.001 would be a very poor model. As for our modeling framework, we had 2 response variables, species richness and animal abundance, and we used several environmental variables. And these we nested within these 2 ideas of biological productivity and the species-area relationship. The first 5, temperature, relative humidity, the presence of fern and moss gardens, a nutrient score, which we scored from 0, no discernible nutrients, to 3, those caves containing the most nutrient-rich resources, the number of entrances, which we hypothesized that this might be indicative of the availability of both alluvial and aeolian nutrients to enter as detritus into the cave. The greater the number of entrances, the greater the likelihood that would happen. And regarding species area, cave length, and cave floor diversity, this 3-dimensional component of how different or diverse is the floor. Does it have a lot of breakdown, a lot of mud banks, sand, etc.? And finally, for the modelers in the audience, we set up the GLMMs using the fixed effects of the environmental variables, and our random effects were the individual cave and the individual study region. And these are the results of the models. First off, I'd like to point out that everything was compared against the null model when we were conducting this model selection process. And because I don't want to see everyone's eyes glaze over, I didn't provide a list of all the models that we used. We provide the best model and then the null model, which is the model that we generated with none of the environmental variables included. You can see the AICC, the difference is at least 0.8 in all cases, which met that criteria of at least 4.0. And also we see that our weight of evidence or our strength of evidence for each model ranged from about 55% to around 80%. Also, I'd like to point out that all variables showed a strong positive influence. They had a z-score of at least 2.0. And that we found that temperature, the presence of fern moss gardens, and that nutrient score that we used were the strongest predictors at both the regional and the broad scale. And also, we found that the variability of relative humidity was a strong predictor of biological diversity for all levels of analysis with the exception of richness at the regional scale. So what we found here through this work is that the biological productivity hypothesis was strongly supported through this analysis. Temperature, variability of relative humidity, the presence of fern and moss gardens, and nutrient scores were those strongest predictors of biological diversity. However, the species-area relationship was not observed. Also, what we hypothesize is that through this multi-scale analysis and through our findings, we suggest that these patterns may indeed be indicative of much broader scale, perhaps global mechanisms driving biological diversity of cave-dwelling arthropods. So up to this point, I want to provide everyone with our conservation implications that we have regarding this work. First off, the systematic sampling, whereby we were employing multiple sampling techniques with multiple site visits, and at the same time addressing the heterogeneity of caves, is going to be critical for maximizing completeness in sampling of a given cave. And also, this is going to be important for establishing baselines for future monitoring efforts of those cave-dwelling arthropods. Also, for resource managers that might be conducting an initial site visit to a cave, we suggest that simply by notating the presence of nutrient resource sites, as well as collecting baseline data on different climate variables such as temperature and relative humidity, could be instructive in at least a baseline information as far as being able to make this quick decision as to whether or not the cave should be temporarily closed or whether or not we should find somebody to properly sample that site. And finally, this is somewhat of a no-brainer, but I still find it worthy of mentioning that for conservation in general, the proper management of the surface environment obviously is going to result in the proper management of the cave environment to a large extent. So in some cases, by not permitting certain activities that might diminish the environmental quality of a given cave could indeed promote the health of the cave ecosystem. The final section of my talk will be looking at cave relic fauna and ecological catastrophes, and I'll be using Rapa Nui as a case study. But first, I want to properly contextualize Rapa Nui for everyone. The pre-settlement environment of Rapa Nui looked much like this. It was a palm-dominated scrubland. We know that it had a maritime subtropical climate. And if you think about it, Rapa Nui was 3,600 kilometers from the coastal Chile and 2,400 kilometers from the nearest island chain. So those animals and plants surviving won the lottery. When they were able to disperse and successfully colonize Rapa Nui, they arrived via a sweepstakes dispersal mechanism. And as you would think, this ecosystem is going to be faunistically and floristically depauperate, and thus likely highly sensitive to both biological invasions of non-native species and environmental perturbations. This is that same caption from the last slide, pre-settlement Rapa Nui. This is an image that I took in 2008. You can see roughly the same view, and you can see that there is indeed a dramatic difference between pre-settlement Rapa Nui and Rapa Nui today. Within 500 to 800 years following settlement by the Polynesians, the island experienced its first catastrophic ecological shift from grassland, or from palm-dominated forest to grassland. Fast forward a few hundred years, the Europeans arrived, and the Europeans converted the entire island into an island-wide sheep herding operation. So today, Rapa Nui is dramatically different than what the Polynesians first witnessed. Today, most native plants and all native terrestrial invertebrates, or all terrestrial vertebrates, have gone extinct. Regarding the insects and kin of Rapa Nui, Dr. Frank Howarth and I compiled a database of the known arthropods for the island via literature review, and we identified at least 424 animals occurring on the island. Of these, and not surprisingly, given that those dramatic catastrophic ecological shifts that occurred, only a mere 5%, or 21 species, were identified as endemic. There was one new species discovery from a cave, Coecobria kinnethi, which was a new species of springtail. But aside from that discovery, there had been no other work conducted underground to address cave biological resources, which was really good news for us. This is a Landsat image of Rapa Nui. This is our study area, approximately 5 kilometers north of the town of Hongaroa. This is a finer scale image of our study area. In red, we have our 10 different cave sample sites. And because we wanted to place these animals that we detected within caves within those cave-specific functional groups that I mentioned earlier, we also sampled the surface by deploying 2 surface pitfall trapping grids. Our cave sampling methods were the same as I described for both the previous sections 2 and 3. And our surface sampling involved deploying 20 pitfall traps per grid, and they were deployed for a total of 10 days. And this is what we found. From this work, we identified 60 species of cave-dwelling arthropods within caves. Of those, 50 were non-native invasive species. We did, however, find 10 of those to be native cave-restricted animals. And we found them primarily occurring within this habitat, within this fern moss garden habitat. Here's a summary table. We had 2 isopods, 7 columbella, 1 Socopteran. Of those, 8 were identified as island endemics. 2 of those are what we're calling Polynesian endemics, meaning that they occur on Rapa Nui, but are also occurring on other Polynesian islands. And interestingly, from this work, we were able to increase the number of known endemics by 33%. We increased that number from 21 known endemic species prior to this work to 31 known endemics today. Interestingly, and you might expect because they did occur within the entrances of these caves, none of these animals were cave adapted. This is 8 of 10 of those animals. And because these animals are cave restricted, this prompted us to examine, well, what are the possibilities for the way in which these animals became restricted to caves? Well, the first explanation is called the climatic relict hypothesis. Now, this hypothesis has been applied to the temperate regions of the world, and essentially, climate change associated with glacial interglacial periods drove species underground. Now, what happened was these surface-dwelling animals were seeking more favorable conditions underground, and several of these animals, or a population of these animals, retreated underground. The surface-dwelling population ultimately went extinct. And over time, that new cave-restricted population evolved into a cave-adapted animal. The second hypothesis is called the adaptive shift hypothesis. Now, this idea was pioneered by Dr. Frank Howarth, who is with us today. And this was his way of explaining why are there cave-adapted animals in the tropics, because the climatic relict hypothesis was explaining what was going on in the temperate areas. So essentially, he explained this by species were colonizing caves to exploit a novel resource. And because caves in the tropics are relatively nutrient rich, there were these multiple opportunities for this type of dispersal and colonization. And what would happen was that as these animals would disperse into caves, ultimately colonize caves, divergence could occur. And the way in which this was explained is that the troglomorphic species occurring within the cave had a closely related congener, or surface-dwelling animal, on the surface. Now here's one of 5 examples that I know of from Frank's work. These are Hawaiian lycosids. On the left here we have the cave-adapted species. On the right we have the surface-dwelling congener. For the cave-adapted animal, this animal has 6 vestigial eyes. The surface-dwelling congener, because it's surface-dwelling, obviously it has 8 functional eyes. But the only difference between these animals was the fact that this animal is cave-adapted. So this brings us full circle back to those 10 endemics from Rapa Nui. Well, we know first and foremost that they weren't cave-adapted, so they're not going to fall into either one of those hypotheses. They don't fit in the climatic relict, nor do they fit in the adaptive shift. So we hypothesized that these animals became restricted to caves via the disturbance relict hypothesis. Essentially, wide-ranging troglophilic animals, animals that can occur both on the surface and within caves, ultimately became restricted to the cave environment due to some ecological catastrophe, such as intense human use of an area. And the way in which we were able to draw these conclusions was first, when we looked at the literature, that 424 record database that Frank Howarth and I developed, none of these animals were identified. So these were new species, clearly. Also, we know that from the surface sampling that we conducted, that they were not detected in any of the surface samples as well. So this led us to believe that these animals were indeed most likely cave restricted. Which brings us to our next question, Polynesian endemics and how they ultimately arrived to Rapa Nui and perhaps to other Polynesian islands. These are our 2 animals. This is a Columbella and an isopod. So there are 3 primary dispersal mechanisms for oceanic dispersal of animals between land masses, the first of which is via wind currents. And for Columbella, there is no evidence of long-distance dispersals between land masses. For the isopod, we have no examples whatsoever as for isopods being part of the aerial plankton. So we were able to rule out this, at least tentatively, as being an explanation for dispersal of these animals. Then is the notion of foracy. Could these animals have hitchhiked with pelagic birds and been dispersed from island to island? Well, there are no known examples of foracy for either Columbella or isopods. Which brought us to our final question. Could they have arrived via rafting? There's a storm, a large piece of vegetation is set adrift in the ocean. Animals are on it as it arrives to a next landmass. If the animals are fortunate enough, they arrive, they're able to colonize and establish a population. We put a big question mark next to this. We wanted to investigate this further. This is a map of oceanic currents for the South Pacific. These are the 3 different islands in which those Polynesian endemics occurred. Our first was the Columbella, and this animal was first discovered on the Hawaiian Islands and recently by us on Rapa Nui. First, I'd like to point out that there's over 7,000 kilometers of open ocean between these 2 islands, these 2 island regions. But more importantly, look at these predominant, very strong cross currents running along the equatorial belt. So we're suggesting that it's most unlikely that those animals were able to disperse from Hawaii to Rapa Nui. When we look at the isopod that was discovered both on Rapa Iti and Rapa Nui, we have 3,400 kilometers of open ocean between those 2 island regions. We do have the South Pacific gyre spiraling in the direction of Rapa Nui. But then when you think about it, Rapa Nui is 62 square miles, it's a speck in the middle of the South Pacific Ocean. So it seems it's a mighty small target for vegetation debris to raft and ultimately land on Rapa Nui. So what we hypothesize is that these animals were dispersed by the ancient Polynesians. Now there is evidence of this, at least indirectly. We know that at least 25 plant species have been introduced to the Hawaiian Islands by the ancient Polynesians. And with that, when you think about it, if you have these if you have cultivars and you're transplanting them if you're taking them from one place to the next, they're going to be in soil. And the Polynesians aren't sterilizing the soil before they transplant their plants or transport their plants. So as a result, whatever's in the soil is going to be introduced to the next area where the Polynesians arrive. And our isopod turned out to be a new species. As I mentioned earlier, it occurred in both Rapa Iti and Rapa Nui. And because we believe that this was dispersed via the ancient Polynesians, we used the Rapa Nui word manu vaca, which is canoe bug, for the species name. And we will be publishing this work. This paper is currently in preparation now. And as for the fate of these 10 Rapa Nui endemics, I would have to say at very best, the future is uncertain. First, Dr. Frank Howarth and Dr. Fred Stone in 2007 reported that the same two invasive species that occurred in the greatest numbers in Rapa Nui caves were threatening the persistence of cave-adapted animals on the Hawaiian Islands. Also, climate change. The IPCC has reported that precipitation in the subtropical regions is expected to decrease due to climate change. And also, Chu et al. In 2010 reported through their modeling efforts that increased drought conditions are expected for the Hawaiian Islands. And also, there have been numerous researchers that have reported that the interaction effect of climate change and invasive species currently threatens the persistence of numerous oceanic endemic arthropod species. However, all is not lost. Because there are 1/3 of those endemic species that occur within caves, I feel that this is a great opportunity for Parque Nacional Rapa Nui to implement useful and necessary management protocols to protect these populations. I've already spoken with the park, recommended that the fern moss garden habitats be considered the most important habitat for conservation of native animals on the island. I've also suggested that they close all the caves containing these habitats until we've been able to at least establish a baseline on the distributions of these animals, as well as just a general idea concerning population size. And all this has not fallen upon deaf ears. Parque Nacional Rapa Nui is in the process of developing the very first cave resource management plan for the entire country of Chile. They are taking this seriously, and they are developing this cave resource management plan specifically targeting those study caves where I've been working the past several years. So when we think about other major catastrophic shifts, perhaps the greatest interplanetary example is Mars. We know that Mars was once a water planet much like our own. It ultimately experienced a dramatic shift and became a hyper-arid place. And the way in which we've been addressing that is, I've often stated, and I've worked with Charles Drost on these projects as well, that if life occurred on Mars, we're going to find evidence of that underground. These are some images from the Earth-Mars Cave Detection Project. This has been a multi-year study where we were developing techniques to identify, to remotely sense caves on Earth, and then applying those techniques to looking for caves on Mars. This is me documenting the collection of remote sensing data when I should have been working on my dissertation. And essentially, the reason why we were doing this is because we wanted to identify the most promising candidates for exploration, because ultimately, in one day, we want to send in something like this. This is me working with JPL engineers this past March. We were field testing a rock climbing robot. And one day we believe that a robot similar to this prototype here will ultimately enter a cave and look for evidence of life. And with that, there are so many people that I wish to thank. Everyone on my committee that Stefan has already introduced for you all. The National Park Services of both the United States and Chile for permitting and supporting this work. Numerous departments here at NAU, the Colorado Plateau Research Station, the Colorado Plateau Biodiversity Center, the Landscape Conservation Initiative, as well as the Department of Biology. Numerous field assistants over the years, and also my funders, the National Park Service, the Explorers Club, and the National Speleological Society. And with that, Maruru Roa for your time, which is Rapa Nui for thank you very much.
And since this is Jut's dissertation defense seminar, there will be an opportunity for y'all to ask questions now, and then when everybody's satisfied, we will break off and have the rest of the defense after that. So I'm sure Jut would be happy to answer any questions that y'all have.
Yes, Tal.
So you mentioned the bugs that you thought were Polynesian endemics, and you were considering whether they came from the Hawaiian Islands. Do you know that, was the ancestor of those bugs from the Hawaiian Islands, or could the common ancestor be elsewhere, and so you're kind of looking in the wrong place for the origin of where those came from, and so if you were to look at a different place, then the oceanic currents might be very different.
That's a very good question. And first, yes, the Columbella is from the Hawaiian Islands. It was first described on the Hawaiian Islands in 1992. It has not been detected elsewhere until our work on Rapa Nui. Could it occur elsewhere? Yes, certainly. And I would think if to follow this idea of Polynesian dispersal of these animals, the next logical place to look, I think, would be the Pitcairn Islands. Because we have Hawaii here, for example, and then we have Rapa Nui here, and then we have the Pitcairns there. And if you're in a navigating canoe and you have nothing but time and you know how to read the water and the land, why wouldn't you do that? That or this. So yes, there is that possibility that we're looking in the wrong place, but because this was the first time that we discovered it elsewhere, other than the Hawaiian Islands, it seems logical at this point to suggest that this is a good explanation.
So there aren't any near neighbors elsewhere? Where do the ones on the Hawaiian Islands come from?
I do not know. And I do not know the genus well enough to be able to say exactly what the origins are of the genus. Frank might be able to provide insight into that, though.
My guess would be the Marquesas, which provide these probably canoes to both. That's considered the origin of some of the Hawaiian canoe plants and animals, and possibly it's the source area of the Rapa Nui, which I believe is considered to be only been colonized a few times. That is the back movement from Rapa Nui to main Polynesia was very short-lived, if at all.
Oh, you're talking about humans moving back and forth. Yes.
Yeah.
Right.
So that limits the canoe bugs that could arrive to Rapa Nui. So yeah, Pitcairn was virtually unknown to the Polynesians as well. If anything, there were only small populations there.
Right.
So that's an unlikely source. As Rapa Nui would be an unlikely source.
I don't know.
Rapa is closer, isn't Rapa closer?
The Pitcairns are about 2,400 kilometers north by northwest of Rapa Nui.
And Marquesas?
Marquesas are going to be up here.
That's the source of the, and somewhat Tahiti too,
Which would be over here.
Would be the source of the Hawaiian material, and probably the source of the Rapa Nui.
Which could track well, also, which is interesting because the Rapa Nui believe that those who first colonized the island hailed from a place called Hiva, which scholars believe are the Marquesas Islands. So that would be interesting to pursue from that angle as well.
There's a lot of convergence between Marquesas and Hawaii as well. And there are a number of bugs in Hawaii that attract to Marquesas, and a few to Tahiti. including another isotope, Australia australis. Southern Polynesia.
Which we have not defined specifically as such yet. Which we haven't defined specifically as such yet. As this idea of canoe bugs.
Well, Polynesian induction, which is essentially some that's where you go.
Yes, Abe.
Find any correlations between any of your species richness, diversities, or endemism indices with whether or not caves are wet or dry, or how recently they've been wet or dry?
We did not investigate specifically how recently they've been wet or dry, but with the modeling, the diversity modeling that we conducted, where we're looking at those different environmental parameters, the variability in relative humidity was identified as a strong predictor for biological diversity of cave-dwelling arthropods. So yes.
But not whether or not they were, so just humidity, but not whether they were saturated or flowing water in them?
No, we did not take those measurements.
Okay.
Any other questions? Charles?
Do Rapa Nui caves have extensive deep zones?
Yes, we identified, well, this is the caveat. With deep zones, because we visited these caves 2, maybe 4 times, definitively saying, yes, indeed, this is a deep zone is going to be a little challenging. So we tentatively said that in at least 4 cases, there were deep zones. But as Frank has pointed out in his paper that he wrote in 1980, he was looking at the variability in lava tube caves. And when there's multiple entrance caves, those deep zone conditions are often not met. And there has to be either a small entrance or an extensive length associated with that cave for there to be a deep zone environment. So we did tentatively identify in at least 4 cases where we thought were deep zone conditions. And we sampled those accordingly.
And no endemic species in those deep zones?
That's the fish that was this big. When I was in one of the deep zones, we were just about to pull the baits, and I saw what looked like a redigid mite, which looked like it had elongated appendages. The antennae looked at least twice the length of the body, and I got down on my knees and started trying to collect it, put it in the vial, thought I put it in the vial, got all the way home thinking, oh my goodness, we have a cave-adapted animal from Rapa Nui, and when I examined the vial, it was empty. So right now, no, unfortunately. Well, thank you so much, everyone, once again. Really appreciate it.